Ahora vamos. Entonces, para esta tarde, la idea es una etapa más en comparación de esta mañana. Sí, no puedo hacer en inglés, bueno, vemos, a veces mejor. Entonces, it's still what to know before doing your analysis, your experiments. And so this morning we were discussing a lot about the databases. That's one point. Okay, I think we won't go back on this point, not that much. And I propose to read two articles that were already on the drive that deals not only with the database, but also more generally about the errors, the biases, and also about the methodological plan, how to sample, what to sample at what scale. There is no like one clear answer to everything, but I thought these articles were interesting. So step by step, we'll discuss it. I think we'll make some break for this. I hope you had some time at least to go through it. They were quite quick. And perhaps they may interest some people more than others. So this morning we already began with this, still with this point. So in the article of Lucy, she was suggesting to know well what are your expected taxa, building a local reference database, so we quite insisted on that point. And perhaps, for example, launching a pilot experiment, having some biological replicates, or including some field negative controls. So those are like suggestions. We'll discuss it, and I will give you some examples also. So to give, short question, but then to detail it. For example, the topic of target taxa, that can be important at the sampling step, not only in bioinformatics. I give you a simple example. For example, for insects, for a lot of mushrooms also, to prepare the sample, we often put them in a paper, like a foil of paper, something to dry them. But paper can contain some tRNA sequences from plants. And so if the metabarcoding targets like eukaryota, you may be over-contaminated with plant primers. Lucy was dealing also with this idea of targeting taxa, knowing what your contaminants are. For example, when she was speaking about the feces to be able to inhibit the amplification of the host or things like this, that's true that if you know well what are your targets, you may exclude some of the products you may use usually. So I often think about paper because we forget that paper contains plants' DNA, and that's a major contaminant in, for example, for fungi, it's terrible because the ITS of plants is sometimes amplified with the common primers. So perhaps think about your products, what you're using. It also means that, of course, you want to be clean and so on, but for example, the dust contains a lot of organisms, so that's basic, but indeed a lot of contaminants comes from dust. And dust, for example, I saw in Mendoza, the Sonda, or this kind of day where a lot of dust is in the air, like you have to prohibit your sampling. But we'll discuss a bit more about it later. The number of replicates, I will insist on the number of replicates, because especially there are big debates today about how we use diversity estimates, if we have to use rarefaction, and this morning, so there were some questions also about the size of samples, the weight. So that's true that to make studies in ecology today with eDNA, we sometimes need pilot experiments, especially to know where we are in terms of diversity estimates. So we'll try to discuss a bit more about this. About the field control, that's also a recommendation. The field control, that may be a bit difficult. It depends a lot of the field. It can be water, for example. We see that we often clean with water and alcohol, water and bleach, whatever, but some water from the field is always a good thing to keep. And so to what I will present now today, it's related to a bit all of these topics, but the idea is really to know your system again, to be able to calibrate the size of your experiments. We'll discuss, I think, a bit more about the coverage, also the number of sequences expected at one point. So just to show you indeed how the sampling also evolved in our team in Toulouse with Lucy, with Julian. You already saw that we often sample at the scale of one plot. We often sample also composite sample. This simple method at the end, which is now used in many articles, but that was presented in Julian Donald, is not like it came from a lot of pilot experiments. For example, Lucy showed you that at the beginning, in like 10 years ago, we began by sampling every 5 meters, then every 10 meters, then every 20 meters. The idea was to say, okay, how can we represent the diversity, knowing that eDNA diversity represent often much more sequences than we were used to when we were, for example, just looking at plants or just looking at fungi. So the idea is to be always higher than your expectation. And for example, you can think it's always thinking that knowing how many species you would expect and trying to have more samples and trying to produce more sequences, at least 10 to 100 more than what you would expect. And then we had also the discussion and the test with Lucy and Julian about the composite, composite or not. For this, there are little kind of all the approaches. When I speak about composite, it's notably because, for example, 1 point here, we now take 16 points per hectare. 1 point is indeed the result of 5 small subsampling, and it's composite. There is a lot of approach of this kind in ecology on transects, circles, whatever, a calicata. There are plenty of composite samples. And why? Because that's true. On one hand, it may reduce the cost of an analysis because you may have less samples. On the other hand, you may have something more representative. It was really good for some analysis where we're interested in the global diversity at the scale of 1 hectare. It was really bad for the questions, for example, spatial questions. So it really, I mean, the composite versus not composite, that's something really you have to think about. So my idea was to show you with this kind of sampling, which is not that high in number and which relies on composite, what are we able to say? And what are the other options that we often see in ecology to represent the diversity? For example, so this approach, we used it, so it's 16 composite sample per hectare. We used it in French Guiana in places where I had absolutely no idea about the diversity of fungi at the beginning. This morning I spoke about the assignation, the fact that we were building the knowledge about their identity. But on the other hand, it was also good to have an idea about the diversity from pilot experiments. So this kind of experiments on a few sites is always useful. Here you see that, for example, I represented in terms of number of sequences, the diversity faced to the number of species detected. The representation in terms of number of sequences, there is a 2 choice. We can represent in number of sites, plots, composite sample, or the number of sequences produced, individuals or here are sequences produced. There is more and more of this kind of representation because indeed the number of sites may be high, low. The importance is to get an idea, a shape of like a species accumulation of this kind. The idea is you want to be, to sample enough, either in number of sequences, either in number of sites, to be able to represent the diversity and to have an expectation of what the global diversity would be. So here you see that on some sites we're quite good, and we're like nearly reaching an asymptote, a plateau. On some other sites we were possibly underestimating. In ecology, there are 2 approaches. Either people would like rarefy and take everything at the lower point. This approach was really used for eDNA and metabarcoding, but it's kind of now really discussed. And so more and more people try to play on the number of sequences produced to be sure to have enough sequences. So in this kind of sampling, my conclusion from this pilot experiment is that we were kind of okay for most of samples, but it was better to have, for example, nearly 1,000 samples per plot. Here, it's represented by plots. So it was better to have 1,000 per hectare, 100,000 per hectare than 50,000, for example, sequences. And if you think about other ecosystems, for example, in vineyards, I see some others companies that are now offering the sequencing for wine producers, and they often recommend also this 100,000 sequences per to represent an ecosystem like a vineyard. In forests, that was something relatively good, at least for fungi. Indeed, this number, the number of sequences and the shape of your curve may really depend on your system also. I must say for fungi, it works quite well. For bacteria, that's something sometimes you may need more. For bacteria, you may expect more heterogeneity also. So, I mean, that's this first check, either on a pilot experiment, either on your sequences to plot the number of species you observe or clusters or whatever, or ASV, at the front of the number of sequences produced is a basic analysis, but it gives you an idea of where you are. If you sequence not enough, too much, that's always a good question to have. One important thing I would like also to remember about this, it's also that indeed to reach this 1,000 sequence, that 100,000 sequence. So I have my 16 plots, but also often I've lost like a good half or like 30% of the sequence. So you always have to overestimate the number of sequences you would like to produce. So why do you lose? Because of quality controls, because short sequences are removed. You have many reasons why you may lose some sequences. So, I mean, never underestimate the sequences. That's something important. Me, more and more, I'm trying to, like, for example, for a specimen, for a database of specimen, I try to have like minimum 3,000 sequences. That would be a red. For a community analysis, if I have several biological plots, for example, 16, I would try to have at least my 5,000, better 10,000 to reach my like 100,000 when putting them. So you have this threshold important. The point I wanted to focus is really that the number of sequences produced is a good way to dimension your experiments. To give you other application of this scheme, indeed, the scheme of 16 plots, like in at 1 hectare, it came from several preliminary pilot experiment projects and so on. We applied it also in places, for example, in temperate forest, where the place where I was mentioning the oldest and like richest forest in Europe. I did, without knowing what would be in soil, I applied the same scheme. Indeed, like here is, for example, one of the curves for the global sites. Well, we still have some more diversity to expect. So here in that case, it may be a bit insufficient, but still it was giving like kind of a high number of sequences, and well, I did not plot it in front of the sequences, but indeed I tried to add for the whole experiments of 12 sites kind of 1 million sequences. So it's again 100,000 sequences per site. And that's something it's like when you don't know your system, that's a good step. On the other hand, 16 plots per 1 hectare, sometimes it's not feasible. So for example, in the Cévennes, in central France, I was also interested in ancient forests in France, in this part of France. But those forests are highly fragmented. Oh, they're not completely fragmented, but they are like neighboring also plantation, and 1 hectare, for example, was too big. So Carolina, for example, that would be your case. So in that case, I tried indeed to make half of the points, like having less transect or adapt a little the number of plots. That's something possible. But on the other hand, when I have this kind of situation, I always try to increase the number of sites to be sure to have enough samples to test indeed the relationship between coverage and the number of sequences. And indeed, for example, so in that case, we had 50 sites, so it was much more, but with a half samples, we were able to compare, for example, the number of expected species, what we call the coverage, and the number of observed species. We see that we were like, when we had a coverage like between 0.9 to 1, it means we were like higher, really kind of oversampling for all of groups. It was for fungi or bacteria. So indeed, in that case, I would say my sampling was perhaps too high. And indeed, for example, more and more in forests in France, a lot of colleagues are just sampling one composite sample, but one from 1 hectare with like 50 points, and they are gathering into one composite sample because they know the coverage will be really high if they produce 100,000 sequences. So this trade-off, though there is no simple rule basically, but the coverage is something I wanted to develop. So about the coverage, indeed, it's not only in eDNA that you would learn about the coverage. So for example, in the group of Diego, with Maria Paola, we read an article at the beginning of this year, which is also from Michael Roswell, published this year, really interesting. I can deposit it on the drive, which is called A Conceptual Guide to Measure Species Diversity. So nothing to with eDNA when you think about it. So the coverage is mentioned in that article. I will detail it. And moreover, for example, some packages are developed to deal with the coverage that are not specially for eDNA. So the important is to think that coverage is not necessarily only for this application. So how is coverage described? The coverage on ecology, at least, it measures how completely a community has been sampled. The idea, for example, is to get the end of a species accumulation curve. So this is the coverage. The end of a species accumulation curve, and the coverage is the slope of accumulation. So, for example, the coverage would be measured by the and would be reached when the end of your accumulation curve is equal to the observed numbers of species observed. So complete coverage would be if you're exactly here at the end of your species accumulation curve. At the contrary, if you're at the beginning of your accumulation curve, you would have a very different coverage. So what we hope, in a sampling, it's to be exactly here. And also to avoid having a rarefaction to do, because that was the other way. So if you want to read more about the coverage, you can read this article. And on the other hand, the big problem with the coverage, and I remember with Maria Paola, that was also a discussion we had with Diego. The problem with the coverage is that unless you have already your pilot experiments, you don't know where you are. So it's difficult to know where you are before doing your analysis. So another solution, when you have already done your experiments and you don't want to do it again, it's also to say, okay, we perhaps have to play with a rare species. Why? Because indeed, what really changes the shape of your accumulation curve is the accumulation of rare species. Indeed, the more abundant you expect to have them at the beginning, and then you don't add so many abundant species at the end, you just add a few rare species. So if you want to avoid having this bias of being there or here, that there is a way. It's called using the Hill number. So Lucy already mentioned it. So we spoke a bit about it this Monday. So if you have never seen a Hill number, don't be afraid, that's simple. So it's a diversity estimate. It's in reality diversity estimates, where indeed it kind of sums the proportion of each individual. So it's kind of a frequency. So it sums all the frequency, but it multiplies also by the rarity of the species. And there is an index here, an exponent, which is the rarity scale of a species. So for example, if we take the Hill and this L is the Hill number. So if the L is 0, for example, you would have the proportion, and the species, the rare species, would have a kind of the same. They would have the same weight as an abundant species. But if we go to Hill number 1 or number 2, for example, you would have a lower importance of the rare species. So the Hill number is playing here on the abundance of rare species, and here again by reducing the number of the abundance of rare species also at the level of the sum. So it has been developed. I mean, it's not a recent approach, but indeed it has been proved to be really efficient for eDNA, notably because with eDNA we have all these errors due to rare species. So Hill number, so you can play with it if you have already done your experiment. So that's true that I could speak about it during the a posteriori analysis, but it's interesting to see that you can play on your experiments on the coverage, and the coverage, it depends notably on your choice. For example, how much you would expect, how much sequences you would like, and it depends also if you cannot do this choice before, it may depend on your analysis later. So in another article, I didn't share it. I saw it recently, and it's a bit more complex, so depends on you, but it's available in bioRxiv. It's another interesting article. It's a group working also on diatoms, on organisms on which the taxonomy is not that known, so it's not like fungi this morning. And for example, they made an article called Decision Making and Best Practice for Taxonomy-Free eDNA Metabarcoding in Biomonitoring using Hill number. So it's kind of perfect if you're interested in what they show notably. So for example, here the D from the Hill number, the 0 or the exponent, you see the small l you saw in the equation. And they show, so I mean, how much, what are the different diversity estimates? And indeed, they try to have different approach. They compared approach, for example, when they rarefied that community, approach when they used the threshold, approach when, so they have different way of defining the filters of metabarcoding. One important point is that indeed they have often similar results, but rarefaction didn't give so many good results. So rarefaction a posteriori is more and more rejected. So that's something important to remember. So and what they show also is that finally, I mean, the Hill number, notably the second, the first and the second, are a good way, for example, to have a good coverage of your sample. For example, here we see that, I mean, we can count the number of samples of the coverage, and it's a way to, I mean, to converge in your analysis. Okay, so this idea of coverage, I don't know if you were familiar with coverage, and moreover, I wanted perhaps to make a small break to see also the point of view of José Reverol, because indeed in the coverage in metabarcoding, sometimes we speak about coverage not only in the ratio between number of species and number of sequences, but from the number of species itself. So I don't know if who is already familiar with coverage in ecology, in community ecology, with sequences or not. Nobody? Okay. But there is no problem with it. I think that so basically, often people are used to make for example, species accumulation curve, like a basic number of sites, number of species. That's perhaps something that is more common. And coverage, indeed, it's an option. For example, if you use eNext on R to make your species accumulation curve, it's just one option more. It's just because before people were not taking care about what the coverage was, and they were more going to rarefaction. Rarefaction often it's more known. So it's a different approach. The good thing with coverage is that it's a way, because indeed the problem before eDNA, we were all, I mean me included, always underestimating the diversity, because indeed we didn't have the sensibility of eDNA. Now with the eDNA, we are like overestimating. You saw the coverage I had in France, for example, in temperate forest, we always have too many sequences, and the coverage is a good way to calibrate. Just to give you some numbers also, I will be back to the presentation, but I wanted to have the feedback of José Riverol, or see if some of you, for example, have already launched an experiment. Do you know how much sequences you had per sample?
Is that a question for me?
Yeah, Jose, for example, for you in your sample, so I know you don't work with ecology, it's more health and so on, but what is the threshold of quality, for example, as a platform? You would say, okay, I don't sequence if I have less than 10,000 or 100,000, or do you have this kind of threshold in your lab?
We do have some thresholds regarding DNA quality, rather than amount of sequences. For example, the OD intervals for quality measurements, and the amount of concentration for genomic DNA. But rather, number of sequences, we don't use that amount.
Okay.
We don't correlate that in health.
Okay.
But I'm taking notes in this.
No, that's interesting. For example, so me, the basic calculation I always do, I know that a MySeq experiments would give me like 25 millions of sequences. I know I would say that half, or I would, after filtering, I would keep like 15. So I would lose like,
okay.
I'm losing.
In that terms, yeah, in that terms, you expect some millions of reads per run, using certain flow cell or certain regions. From what you expect, the quality may cut that from 1%. It's around 4 or 5%, which is expected, like 95% of quality reads. You would get from that million reads expected, you know. But we don't speak in measure of a threshold. We want that coverage. We understand coverage as another definition. We understand coverage as how many reads you get per alignment in one position specifically.
No, but that's why I wanted your point of view, because indeed I know we don't speak about coverage in the same sense, and that's important. So that's why I was wondering. And for the others, that's always good to know, because notably in platforms, for example, you can send a wine producer could send one sample and say, okay, I want a sequencing, but it's pooled and you never know how many sequences. Me, I always try to know how many samples I would put in one experiment. And for example, if I know that from my 100,000 sequences I would expect, 25 million sequences I expect. If I know that I would keep like half of the sequence clean, I would say, okay, I don't want more than 2,000 specimens in it, because if not, I would be lower than my threshold I have on the coverage and number of sequences. So it's just with, I didn't make an exercise on it, but always try to know what number of sequences you want for your experiments. For example, if you're making an experiment on 3 species, okay, 1,000 sequences would be okay. if you're making an experiment on marine biology where you don't know how many species you have, or for example, in Patagonia with Carolina, we don't know the number of species, always try to be higher so that you be sure you will have a higher coverage and you be sure to be able to make good diversity estimates. So that's why at least sequencing like a bit higher, it's always important. Okay, so back to the, well, I'll try to make some steps like this. So to be back in forest and then discuss a bit still about the sampling plots. So indeed, as I mentioned, the sampling grid we had with Lucy and Julian, we applied it with different colleagues. The idea was also to like have a common observatory. There was, for example, a project involving Carolina that was deposited in South America, also to make something similar. The idea was to test also how many samples do we need to represent diversity. We all began with something really extensive, like for example, the 15 to 16 number of plots, composite often, always on 1 hectare. The 1 hectare was not discussed because it was in relationship with forest estimates, dynamic of forest, number of species, and so on. So we never changed the 1 hectare, but it was more the density of plots within the hectare that was changed. And what we saw also is that one important thing to represent the diversity, at least regionally, it was perhaps much more the number of sites. For example, we are not necessarily homogeneous in the number of sites. In French Guiana, there were 50, in Colombia, 20, in Patagonia, 4. In Canada, I have 11, some other plots there. So indeed, the number of plots was different. But as you saw yesterday with Guillaume Schwab, that's really important to have distant sites, and the number of sites may allow you to interpret and to analyze your signal. So I didn't show any result from this large-scale experiments. Indeed, it's ongoing. But I will also show you that, I mean, that's what we did in our team, but there are also different ways, and it really depends on your system also. For example, before the GlobeNet experiments through the PhD of Mélanie Roy, so my former student in Canada, we had to study also a place where nobody had studied the fungi before, but where we had some big hypotheses on the weight of fungi as a facilitator of the fur seedling. Indeed, in the condition of climate change and climate warming, we see that fur are increasing their distribution north, so they're changing the dynamic of forest there. And there are 2 kinds of forests where fur can grow. We see that they can grow under some conifers and some broadleaf trees. And we're always seeing like the fir more associated with a broadleaf tree. So the idea was perhaps they may share some fungi and so on. So anyway, it's a biological question, but we had this question on soil. And in that case, it was a bit before. Indeed, it was published in 2018, but it was done a bit before. The idea was that it was kind of a pilot experiment. So with a pilot experiment, what did we do? At that time, I had less money also to make my experiment, so I was not with my 15 or my 16 plots per hectare. So we did 5 transects and we pulled them, like having 5 pseudo replicates per site, because they are pseudo replicates, then that's a kind of tolerance. So we had these 5 composite sample per site and 4 pairs of sites, and indeed, and 2 horizons of soil. We had some striking results in terms of viability of, for example, the different plant species, host plant species had really different soil communities. We often see a difference between mineral and organic horizon. But I must say, we were really lucky because I think 5 was really the minimal variation. And we saw a big variation because also the soil, you can see the organic and the mineral horizon here, the soil was extremely contrasted. And broadleaf versus conifer trees were extremely contrasted. So indeed, the conclusion of this example is that you can play still with the number of samples. It always depends on the heterogeneity you expect. if you have a huge heterogeneity and big differences between ecosystems, okay, you don't need so many samples. The case of 16 samples was a bit too much, I think, in many cases, but it applies in systems where you don't know what you expect, and at least you expect you can overestimate your diversity. I mean, oversample your diversity. Okay, so that's an example. Another examples, right now in Argentina, I'm also working with vineyards. And indeed, vineyards, it's a completely different literature, much more in agronomy, less in ecology. The interesting thing is that there were kind of pilot experiments, for example, at the worldwide, like 50 countries. There were big experiments testing a bit the scale, at what scale shall we sample and so on. And in vineyards, because the heterogeneity is kind of low, you don't expect so many variation, everything would be extremely local. The map is completely different. We make five sampling that we pull in one composite sample, and normally it's enough to describe the diversity of like a 30 per 30 meters plot. But so on one hand, it's good because it means, okay, we expect a high coverage because we will sequence a lot on a small amount of species. We don't expect as much species as in a forest. But on the other hand, me as an ecologist, I mean, applying this kind of protocol, for me, it's a bit like a thread because I don't have the replicate, the viability. So what do? I increase the number of plots. For example, I try to sample 3 different plots per bodega, having 160 plots globally to make the study and to be able to make some models and to be able to make some statistics. So it's another way of playing. It's a bit like what yesterday Guillaume Schwab was also suggesting. You can have few samples per site because sometimes with a few sample or a few composite sample, you may represent well the variability, but you may increase the number of sites. And so to choose between these approaches, you have to know your system, have to know a bit the idea of the variability, spatial, temporal eventually. That's really important. So of course there is not a clear answer, but the articles I was suggesting, like to make some more choices, are important. For me, it's really a play and a trade-off between what you would like as a biological replicates. So there are some guidelines, of course. For example, the project's GlobeNet is often suggesting the 1 hectare, the composite plots. You have a lot of worldwide initiatives that are suggesting some methodology. So, but think about your experiments first. That's important. Then you can play on coverage. So coverage, it can be the number of sequences. And that's something we are not used to play with. It can be saying, okay, I will try to get 100,000 sequences from one sample. And it may give you a good result. With only one sample, you may have like a diversity estimate anyway. So, and to play with, the big problem is that as eDNA metabarcoding is, it's cheaper, but it's not cheap now. You may play, it's what I do in my research also, I play with the number of samples I can afford, the size, the sampling size, and if I need to increase the sampling size, I use composite sample. As soon as I don't have any question on the spatial distribution, because composite sample, it's a bit problem. Okay, so that's like some advices. And on the other hand, speaking about, it's a comment about one of the discussion this morning. That's true that if it's for a question of community ecology, in that case, we spoke about the weight, how to calibrate. That's true that surface weight can be used to calibrate, not for specimens, but more for perhaps for community ecology. It's possible and not. I mean, there are 2 sides. For example, for soil, I know that my calibration is made based on the number of species expected, but also about the DNA extraction method that I use. For example, I know that for my DNA extraction kit works well for 5 grams of dried soil. So I never sample more than 15 grams of dry soil because I know it's my experimental limit. And on the other hand, it may be enough to represent to have 500 species. So you always have to know also some of the experimental limits you may have, or depends again about your question. For example, I often work at the scale of 1 hectare because I know that my fungi, some are larger than 1 hectare, but I would expect to have several, my individuals included in 1 hectare. So I think about the size of my fungi. I think about the dispersal. Most fungi are dispersed like locally, so 100 meters in distance, I would be okay. But for example, if I have to replicate my number of sites, I would take into account the dispersion of species. And so this kind of knowledge, you know it's not particular to eDNA use, but it's important to calibrate a bit your sampling. And so to conclude with this part, the number of sites, Guillaume was also mentioning it's super important. And so that's why, Carolina, we had some discussion also. It's always a trade-off. I always try to have for one experiment kind of 100 samples, either distributed on like 10 per 16 per 8 sites, or having a minimum of replicates and increase possibly the number of sites. Okay, we'll shift to other topics still about the sampling. Are there some reaction now? Or some discussion, or just to know a bit the scale, at what scale you're, or if calibration, if some have used some calibrations this morning. We spoke about calibration and weight. Are there some trials already to calibrate the sampling? No?
Mélanie?
Yeah.
I have a question.
Yeah.
Can I speak in Spanish? Because
sí, bueno, sí.
It's easier for me. But okay, mi caso es un poco diferente a estos ejemplos. En el caso de hacer ADN ambiental con muestras de agua.
Sí.
Es diferente por varios motivos. Por un lado, en los ríos el agua se mueve. Entonces estoy integrando muestras de varios sitios del río, y además tengo la diversidad, obviamente, que es mucho menor. Estamos hablando de 15 especies como mucho, ¿no? Pero mi pregunta ¿es una estrategia posible mezclar muestras de diferentes sitios en el caso de muestras de agua? Tal vez en el suelo también. O sea, si yo solo quiero saber la diversidad de un río, ¿puedo mezclar muestras de diferentes sitios de ese mismo río? Y en ese caso, ¿qué conviene? Si mezclar el agua, mezclar el ADN extraído, mezclar los filtros. ¿Sí?
Y, depende siempre de tu pregunta. Si...
You can speak in English.
No, en castellano está bien, de hacer una pausa en castellano está bien. Y mira, entonces yo no trabajo sobre agua, pero colegas de mi laboratorio trabajan sobre agua, y es verdad que yo creo que a veces usan el mismo filtro y acumulan diversos muestreos en el río. Así hay solo un filtro que es tipo con 10 horas de muestreo en diversas partes. Creo que es una estrategia, porque mezclar el DNA es también posible. Por ejemplo, si vos pensás que con una extracción de DNA ya estás al límite de la extracción, porque tipo hay un límite, el DNA, tener demasiado DNA para una extracción no es bueno también. Por ejemplo, para mi raíz esta mañana, para mi raíz nunca hice una extracción con más de 50 pedacitos de raíces, porque si no tengo demasiado taninos, otros problemas. Así que se puede también mezclar el DNA. El problema es también la cuantitatividad, de decir, bueno, ¿qué tengo después? Pero si una pregunta es, por ejemplo, detectar una especie invasora, una presencia-absencia, lo puedes hacer sin problema. Y también la otra posibilidad que se hace en diversos laboratorios es de decidir si vos haces un muestreo separado, una extracción separada y quieres hacer una amplificación separada, puedes usar tipo mezcla de primers que son con algunos bases de diferencia y después hacer la mezcla y solo hacer el sorteo bioinformático. Yo, por ejemplo, hago mis 16 puntos que son compuestos en una parcela, pero hago la PCR separada con primers, con algunos bases de diferencia y mezclo todo al fin. Así tengo mi mapa de mi curva de acumulación y veo bien dónde soy, pero al final, para, por ejemplo, los índices de diversidad, lo calculo siempre al nivel de la parcela, de un hectárea, haciendo mi mix. Pero porque también me gusta tener un poco de información sobre cada punto, sobre la distribución espacial. Pero si no buscas eso, sí, mezclar es posible. Es solo que para la cantatividad después no ves la diferencia. Y después yo uso bastante muestreo compuesto de este tipo a una escala pequeña, es verdad, porque también, por ejemplo, si yo lo veo con los microorganismos, el efecto del pH es súper importante. Así que si a un lugar hubo alguien, no sé, un colega que hizo un pis o algo, no sé qué, no sé, un organismo, un cacao, no sé, cambiar completamente la comunidad de microorganismos. Y no quiero este efecto súper local. Así que por eso uso el compuesto que me saca un poco esta super variabilidad local y no lo veo la diferencia. Es verdad que es como un sondeo en una población. Si haces el sondeo global, al final tienes la abundancia global y es bien, hay efectos locales, pero si haces, por ejemplo, en tu muestreo de agua, haces 10 veces o 30 veces el muestreo, al final la abundancia sería bien estimada a un nivel de un río y te cuesta menos en número de muestreo y al fin tienes algo bien representativo y mejor que hacer 30 muestreos que te cuestan un montón y que al final te dicen poco de la diversidad. Así que, no. Pero puedo, si quieres, te paso algunos contactos porque tengo colegas también argentinos en mi laboratorio de Francia que trabajan sobre diversidad de pescado y usan el agua desde en Guyana francés y no sé, no creo, no trabajan en Argentina, pero trabajan mucho con el agua ahora. Y puedo preguntar, yo de lo que me recuerdo usan el mismo filtro en diversos lugares, así se llena el filtro y es un modo para usar solo un filtro y muestrar todo un río.
Bien. Y otra pregunta que mencionaste, lo de los taninos. ¿Qué experiencia tenés con los kits que ya vienen con columnas para extraer inhibidores de PCR? Si vale la pena el gasto o se puede jugar más con lo que son las concentraciones y demás para que las PCR funcionen.
Bueno, ahora, para eso depende mucho de tu modelo. Por ejemplo, me recuerdo en doctorado, yo siempre trabajé con hongos, tierra, suelo, raíces, cosas que son tipo en zona super alta diversidad, con 30 especies diversas. Y a un momento hubo una empresa que quería vender mi prueba de inhibidor y todo eso. Y al momento renunciaron porque era demasiado test para bajar el nivel del manioc, bajar el nivel del... Así que demasiado fuente de taninos. Bueno, pero de un otro lado lo veo con el suelo. Antes para raíces no usaba el kit, porque para raíces al final, como vos decís, yo tenía tanta diversidad de taninos y al final funcionaba mejor de tomar un poco de tiempo para bajar el nivel de magnesio, jugar con la bovine cereromalbumin, jugar al nivel de la PCR, es verdad. Con el eDNA ahora uso kit. ¿Por qué? Porque de una manera cuando hago una amplificación, es verdad que finalmente no necesito mucho producto PCR, así que al final cuando no veo un producto PCR no significa que no hay sobre un gel, por ejemplo. Así que hacer la prueba, testear y esperar la secuenciación para tener el resultado, yo no puedo, pero porque es demasiado... tengo que abrir la puerta, pero sigo respondiendo sobre eso. Entonces, uso kit por dos razones. Veo que hacer test de mi amplificación, verificar con la secuenciación, me cuesta demasiado tiempo, porque el tiempo de enviar un muestreo, de decir, bueno, tengo una respuesta o no, no es como una PCR normal, donde esperas tipo un gel y tienes tu resultado. Cuesta más, así que no hago eso. Y además, lo vi sobre el suelo. Al final hubo buen desarrollo de kit que trabajan con el suelo, así que al final con el suelo no hago tanto test. No sé para el agua, pero para el suelo ahora uso kit. Ayer envié, no lo mencioné a Carolina, pero tipo media hora después el curso recibí mis 500 muestreos de kit, así que es posible en Argentina. Yo encontré los kits que me gustan bien para el suelo, con biocientífica, así que bueno, después hay diversos, ¿no? Pero es posible cuando uno compra bastante, de bajar los precios y todo eso, y al final vale la pena, porque si una columna te cuesta $10, y si al final mandar un muestreo para hacer un test y saber si funcionó o no sin kit o con kit, te cuesta $1,000 porque haces 5 muestreos y cada uno te cuesta $200 a la secuenciación, no vale la pena. Así que al final ahora uso kit. Y eso vale para Carolina porque lo hablamos también. Es por eso, porque verificar si funcionó sin ver bien la diferencia sobre el gel, sin saber un poco también porque es más comunidad, ecología, así que preferisco usar tipo un poco de seguridad y trabajar con los kits. Pero, última respuesta, cada vez que hago una amplificación, uso primero el DNA, una dilución del DNA, por la duda. Siempre uso bastante replicates, tipo a la PCR, para bajar un poco este efecto de inhibición de la PCR o problema que podríamos tener. No juego mucho con el magnesio y con la bovine serum albumin, porque en general con la dilución del DNA primero funciona, pero hago la dilución por la duda, replicates, y al final, bueno, a veces tengo un muestreo que no funcionan, es verdad. Fue el caso, como yo lo hablé esta mañana con specimens, pero en general son tipo viejos hongos o cosas contaminadas. De muestreo de suelo, con esta técnica, con kit, en general no conservo todo hasta el fin. Hay un poco de contaminación, pero poco, y no funcionó bien. Así que, bueno, no es para vender kit, pero bueno, es una discusión interesante porque lo hablemos también con Carolina recién, si puede ser mejor o no sin kit, y vale la pena si hay un kit que existe para algo. Y, bueno, no sé, y entonces para tú saber un poco, vos, Tomás, ¿usas kit? ¿Qué haces? ¿O estás...?
Sí, hemos usado kits y sí, funcionan mejor. Acá tenemos, con el tema del agua, depende la época del año, la estación del año.
Sí.
Los ríos en primavera y verano tienen mucho material en suspensión, y ahí sí está bueno tener los kits. En cambio, en invierno y en otoño el agua es transparente, no hay material en suspensión, entonces es mucho más pura el agua.
Okay.
Y ahí, bueno, ahí Cristina tiene la mano levantada, ella es la que lidia con las PCR, pero sí, por lo general lo solucionamos con diluciones.
Ok, bueno, lo
Diluyendo
mismo.
mucho la...
Ok, Cristina, sí, dime, decime.
Sí, Melanie, dijiste dos cosas súper interesantes que quiero constatar a ver si entendí bien. Por un lado, mencionaste que vos amplificás de tu muestra ambiental con primers diferentes y después juntás los amplicones para enviar a secuenciar.
Sí, es eso.
Bien.
Ah,
¿Eso lo haces para aumentar lo que podés captar en esa muestra ambiental?
no, ahora se podría hacer en este censo, por ejemplo, hacer bacteria, hongos, ¿no? Pero después hay el problema de tamaño de la secuencia y el problema de hacer la secuenciación en el mismo lugar. No lo hago por una pregunta básica. Pensé hablar de eso este lunes, pero bueno, básicamente cuando uno envía su producto PCR a la secuenciación, en general, para la preparación de la librería, para agregar los adaptadores que son al fin de la secuencia, hay otras PCR que son hechas y pueden ser fuente de errores. Así que a un momento, la idea era de mi laboratorio y el laboratorio de Lucy también de no usar los tags al fin o de usarlo en ligación sin amplificación. Así que es también la propuesta de otros laboratorios de juntar amplificación con primers que son, pero solo una parte de diferencia, tipo 3 nucleótidos o 4 nucleótidos en forward y reverse, y te permite de reconocer tu amplificación, de decir, bueno, eso era el muestro A, y el B tiene un otro, y el C un otro, son combinaciones, y te permite con la bioinformática después de buscar tu muestreo, te permite de aumentar el número de muestreo por library. Hay límites a eso, porque, por ejemplo, si tus primers son demasiado parecidos, después hay un poco de chimeras y todo eso, pero es una propuesta. Y eso hay varios laboratorios que lo usan.
Bien, perfecto. Y la segunda es que vos mencionaste que a veces no obtenés una banda muy potente o no ves banda e igual mandás esa muestra a secuenciar. ¿Puede
Sí.
ser que dijiste eso?
Sí, lo hago.
Porque
Y porque, mira, el gel, hay que imaginar que con el gel ves cosas que son tipo, no sé, con 5.000 de reads o algo, una PCR súper potente en general. Y, por ejemplo, si hablo si busco, por ejemplo, la secuenciación de un espécimen, por ejemplo, mi espécimen de herbario. Esta mañana vimos ejemplos con mi espécimen de herbario. Con el Sanger, con la PCR normal, nunca funcionó. ¿Por qué? Porque la PCR, imagino, era demasiado liviana, porque había un poco de contaminantes, porque el ADN era ya un poco destruido en herbario, así que era pedacito, así que el uso de marcador corto funcionó. Y también envié a la secuenciación para saber, y había secuencia, había pocos, pero había. Así que yo hice mi sorteo más con la filogenia, con base de datos, para decir, bueno, eso es la secuencia que busqué, no es una levadura, es mi uracilase, y lo vimos esta mañana, puede funcionar. Depende un poco de tu interés, pero por ejemplo, para espécimen de herbario, mis colegas que trabajan más en conservación mandan muestreo donde la PCR no funcionó bien o había contaminantes, lo mandemos y manejan con la bioinformática a identificar las buenas secuencias.
bien, perfecto. Porque yo me estoy rompiendo la cabeza básicamente para obtener una buena banda en la PCR y no lo estoy logrando. Veo una banda, pero tenue, y por ahí es suficiente para mandar a secuenciar.
Sí, podemos preguntar a José Reverol, pero nosotros se puede enviar para mí. Lo sé que, José, decime, sí.
Usualmente en humanos también suceden ese tipo de cosas. A veces no ves, porque aparte correr un gel también es parte del control de calidad antes de enviar muestras en humanos. Corres un gel para ver DNA genómico, la presencia como el control de calidad de tu extracción. y muchas veces está tenue o muchas veces está borroso. Pero dependiendo del caso o la urgencia médica que tenga la persona, de todas formas se envía y uno depura toda esa data que viene después, posteriormente, con la bioinformática, como dice Men. Con la ayuda de la bioinformática depurás, a veces, no sé, te sacás encima del 40% de la información que es toda basura, que es muchísimo, pero a veces con el 60% que te queda, te basta como para interpretar resultados.
Perfecto. Y la última pregunta que tengo es sobre la definición de cobertura de la que hablaban antes. No en los términos ecológicos, sino en el término bioinformático. Si yo tengo un sitio como el que mencionaba Tomás, que sabemos que tiene baja diversidad de peces, ¿es necesaria tener un número tan alto de lecturas o yo puedo poner más muestras sabiendo que la diversidad es menor? No sé si se entiende la pregunta. Por ahí es
Sí,
más para José.
sí, sí, es una buena pregunta. Yo tendría que decir, bueno, por ejemplo, una cobertura de 3.000 o 5.000 de reads es nada en comparación de lo que hay pero, por ejemplo, el Chile, no sé para tu plataforma, José, pero pregunté eso a la plataforma de la mayor al Chile y el precio era diverso. Yo decía, bueno, si quiero muestro, por ejemplo, un espécimen, quiero 10.000 secuencias, bueno, me costaba menos que si quiero 1.000.000. Es el número y así que si vos quieres saber lugares donde hay pocas especies y estás bien segura de tu muestreo, es que puedes bajar, obvio. Es solo que para mí es bien de tener tipo 3.000, 5.000 secuencias, es también no únicamente para tener tu estimate de diversidad, pero también para sacar los contaminantes, porque lo menos DNA, así, lo menos DNA, lo más contaminantes puedes tener, porque si hay poco DNA, por ejemplo, un polvo, un semilla de polen, bueno, no sé, el polen se amplifica más que tu DNA. Así que sí, puedes bajar, pero siempre tener en cuenta que vas a sacar los singletons, todo lo que es contaminantes, y si tienes pocas especies, bien de tener un poco más tipo de concentración de DNA en este caso. Así que hay que jugar, pero
Un
bueno,
detalle que tampoco se toma mucho en cuenta, Melanie, y que quisiera agregar también a la serie de factores que te pueden afectar la calidad final de tu muestra, por eso es que se hacen tanta cobertura, digamos, por RIC, a pesar de que sea corto, es que aparte, por lo menos Illumina, y también pasa con el Oxford Nano por el Minion, la síntesis en Illumina, en el caso de Illumina, es por síntesis, se hace en PCR. Entonces ahí también acumula errores. Y hay errores de fase y todo lo demás que probablemente lo veamos el lunes. Igual en el Minion también ocurren errores de detección.
Bien, bien. Perfecto. Gracias a los dos.
no, bueno, entonces, bueno, entonces es bien de charlar de eso porque típicamente sí, el señal sobre el gel, yo siempre verifico, bueno, yo, no, los ingenieros que hacen la especial conmigo, porque no hago todo, pero bueno, hacen una verificación para saber, por ejemplo, si recién hubo un experimento donde nada funcionó, así que no lo vamos a enviar. Imagino que hay otros problemas y lo vamos a resolver, pero si algunos no funcionan no sé, sobre una placa PCR, bueno, lo mando igual y vemos. Y en general, si depende si es para un espécimen, en general hay DNA. Y si para es una comunidad, bueno, si el coverage al fin es bien, está bastante, está al final, bueno, ¿por qué no? Lo mejor es de tener bastante biological replicates o lugares, de aumentar un poco su número, porque es verdad, es el problema del eDNA, es más sencillo, así que hay más contaminantes, perdimos bastante muestreos, secuencia, así que hay que aumentar un poco al momento el número de muestreo. Bueno, si no hay otras preguntas, vamos a volver a la presentación. ¿Dónde no? ¿Dónde es? Estuvimos acá.
Perdón, una consulta.
Sí.
Perdón. Nosotros en el laboratorio lo que hacemos es extracción de ADN con kit, hacemos extracción de ADN de suelo completo.
Sí.
Nuestra idea es mandar a secuenciar ese ADN extraído. La idea es empezar con bacterias presentes en suelo, no un género ni nada en particular, sino ver qué es lo que tenemos, tipo un metaanálisis, digamos, de lo que hay primero. En el caso de nuestros muestreos, digamos, nosotros hacemos un pool también, tomamos diversas muestras en los suelos y hacemos un pool, de ese pool extraemos el ADN y eso es lo que queremos mandar a secuenciar. Son dos las consultas. ¿Cuál sería el número de réplicas a mandar a secuenciar de ese pool que me permita a mí tener un resultado bien confiable, si es que hay que hacer réplicas? Y el segundo, este porcentaje de cobertura que tiene que tener el análisis, digamos, en el caso de tanta diversidad. ¿Cómo puedo estimar que es un buen porcentaje de cobertura, teniendo en cuenta que solo quiero ver, a lo mejor, bacterias? ¿Se entiende?
Bueno, sí. Alors, dos cosas. Te hablo de la segunda primera.
Sí.
Lo mejor, y vuelvo a mi presentación, lo mejor es devolver a este tipo de curva. Si puedes, con tu número de especie de bacteria, saber si tienes una curva y eso se hace, entonces no necesitas, por ejemplo, con un muestreo,
Sí.
puedes hacer este tipo de curva. Lo que hace eNext va tipo muestrear aleatoriamente, tipo, en orden aleatorio, en tu muestreo, en tu comunidad, y construir este tipo de curva. Y lo hace 1000 veces y al final, con un muestreo, puedes tener este tipo de curva, con la acumulación de cada especie y el número de especies que tienes al final. Entonces, funciona con... si tienes, por ejemplo, yo tenía mi 16 por puntos, pero con uno funcionaría también para testear este tipo de curva. Entonces, en tu caso, yo haría eso, si tienes ya un experimento piloto, cuando tienes tu resultado, para ver dónde estás. Eso es importante. Bueno, ¿por qué no veo? Y la otra pregunta era sobre el número de pool. Yo, por ejemplo, por un pool, yo solo uno, pero tengo diversos pools también. Entonces, por ejemplo, tenía acá, mandé mis 16, entonces cada uno es un pool y hago solo uno de un pool. Conservo un otro al freezer para la duda, pero yo solo uno por pool. Y como tengo eso, para mí es bastante. En el caso, pero eso era en el caso donde, por ejemplo, en Guiana Francés, yo no puedo volver al campo, es súper complicado y estudié 10 lugares. Y gracias a la, tipo, 10 por 16 me hace 160 y puedo hacer un artículo y responder a algunas preguntas, pero es súper limitantes. Lo que hago ahora, por ejemplo, en viñedos, en bosque, donde puedo volver o agregar más muestreo, pruebo de tener tipo, un compuesto global, enviar uno a la secuenciación, pero tener mucho más puntos. Así son más replicación biológica o ecológica. Así que para vos, o tienes una pregunta súper local y entonces necesitas, tipo, como en estadística, 30 muestreo para tener algo representativo, o tienes 30 sitios. Es una pregunta más global, no sé. Y yo en tu caso, si ya enviaste algo a la secuenciación, la primera cosa sería de decidir, bueno, dónde estoy en la curva y en general con bacterias, si hiciste una librería por librería, es tipo súper, es el top. Tipo, en general, la las plataformas prueban de secuenciar bastante. No sé.
Sí.
Así que...
Bien, gracias.
No, pero si hay otras preguntas, lo podemos charlar, ¿no? Pero es siempre un compromiso también con, yo lo veo con el número de factor que testeas. Por ejemplo, si testeas solo, no sé, agricultura biológica convencional, tipo dos cosas, bueno, 15 en 15 ya tienes una comparación, o mejor 30 y 30. Si agregas otros factores, bueno, hay que aumentar, pero no es únicamente el eDNA que te permite de decidir eso, es también tu nivel de variabilidad, tipo del ambiente. ¿Sí? Bueno, cuando recibís tu muestreo, haces tu curva y listo. Bueno, ¿seguimos? Up. Entonces, estuvimos allá, volvemos. Una cosa importante también para terminar sobre este punto es que estas preguntas son exactamente las preguntas que me parecen interesantes a tratar en el proyecto de cada uno, de ser capaces de decidir, bueno, para mi pregunta, ¿cuántos muestreos, qué protocolos? Es eso un poco que para mí el ejercicio, el report que me gustaría que cada uno haga para este curso es eso, es de tener su pregunta sobre qué hay que hacer antes en base de datos, en plan de muestreo, en número de muestreo a tomar, de coverage, de compuesto o no. Son todas preguntas súper importantes a pensar antes de tener resultados. Bueno, y después vamos a seguir y discutir un poco del artículo que mandé, pero para discutir si tenemos ya las preguntas sobre la escala el tamaño de, bueno, voy a volver en inglés. So, if we have so clear ideas about at what scale we would like a sampling, the database and so on, now I think we are kind of ready for sampling. So for sampling, indeed, there are different protocols. Myself, indeed, I should clarify also all the protocols. The idea is always to be the cleanest possible. So for example, a good colleague, Christelle Martineau in Quebec, she's involved in many like microbiome projects. She's always suggesting to clean with ethanol. That's something, I mean, me, I must say for DNA, I like using the bleach because ethanol, the problem is often dehydrates the DNA, and so DNA can stick to your metal and whatever you use. And for example, root cores and so on are really bad with to clean with ethanol. The DNA sticks to it. So I use bleach, so that destroys the DNA. So the better is 1 minute bleach. That's even better. So the protocols may differ, but in general, the cleaner, the better. Plastic bags that are clean and so on. And so I really warn you about your tools. For example, the soil cores. Some colleagues use the soil cores, but soil cores are extremely difficult to clean. So you have to think about how clean your material are. It's really essential. And so for this kind of bias guidelines and so on, I've invited you to read the article from Liu from last year. That was kind of interesting because it was discussing all the bias, the contamination, also the procedures of calibrating, and I thought it was kind of interesting. So, and for example, we'll discuss it a bit later, but for example, they were interestingly showing how people are decontaminating, for example, their tools. So it really depends of the people. So it was about insect from aquatic environment. A lot of people are changing equipment. So for this, depends of people. Me, I try to recycle what I could. So I try to use bleach and recycle what I could. But of course, for example, plastic bag, I never reuse it. I think that we should not reuse. Some people are washing. So after you have to think if you wash with soap, ethanol, bleach, all three. Some people are sterilizing, which is good, but time consuming also. You may see that some are not doing anything and that's bad. But anyway, and then you have to think also about the preservatives, but it's not only abuse for eDNA. For example, in this article, she was mentioning a lot of ethanol. For me, ethanol and tannins are making a bad mix, so I never use ethanol. So I either dry in silica gel, as Julian said, or freeze. And you see that, I mean, there are different methods always. the importance is really to think that you work with short sequencing or sometimes long sequencing. That's true. So you want to preserve more the DNA than organisms themselves. And for example, freezing, defreezing DNA is extremely sensible. So I pay a lot of attention to freezing, defreezing cycle, a lot of attention also to ethanol, because ethanol may stick my DNA. So I more use bleach and plastic surface, everything that can be easily washed also for this. But for this, I think this article was nice to present the different aspects. So I wanted to know if some of you have read also this article or found something interesting. I know that some people in the group are interested in insects, so I would just quickly discuss, know if some people have read it or had something to add from these articles. Nobody had the time to read it, but perhaps I apologize.
Yo trabajo con insectos.
Sí. ¿Qué pasó?
Sí. No, no leí el artículo, pero sé toda una serie de pasos que uno tiene que seguir para llegar a tener un resultado confiable, como lavar los insectos, no sé, por ahí para las chicas les sirve a las chicas de polen.
Ah, ok.
Hacer un lavado superficial y todo ese cuidado de estar, si se hacen disecciones, dónde hacerlas, por ejemplo, no es lo mismo hacerlas en PVC que en agua, y tener mucho cuidado con los elementos de disección también para llegar a tener resultados confiables. Y también es muy importante, al menos en el caso de insectos, cómo se preserva, porque sí se han visto que a veces los resultados distintos no son comparables los microbiomas. Si el material para preservar la muestra hasta que se extrae el ADN es diferente.
Sí, no, y eso son cosas básicas, pero sí, yo lo veo un poco también en el lado por ejemplo, para estudiar hongos que viven adentro de una hoja, de los endophytes. En general prohíbe de esterilizar más con alcohol. Ahora esterilizo también con lavandina para destruir el DNA que está arriba. Así que hay que pensar siempre DNA más que organismos. Es un poco un pequeñito cambio, pero es importante. Y es súper importante de ser súper limpio. Nosotros lo vemos, voy a compartir una foto del laboratorio el lunes, pero de tener miedo de ventana, polvo, vestidos, todo, porque son fuente de DNA. Sí. Así que para eso, bueno, lo vamos también a hablar este lunes, imagino, un poco más, pero con la parte del laboratorio. Y además no hago también un curso, la parte del laboratorio es un poco difícil a compartir por un Zoom, pero bueno, ser súper limpio y pensar siempre al aspecto DNA. Y después hay el número de control.
Sí, desinfectar mesadas también.
Sí, no, pero eso no es tan específico del eDNA, pero hay que tener más miedo que para un producto normal. Bueno, entonces no sé si nadie leí este artículo, bueno, era solo porque me parece interesante la parte guidelines, de decir, bueno, a qué etapa tenemos que tener un poco miedo o tener en cuenta los contaminantes. José, ¿alguien quería compartir algo o no? ¿O seguimos? ¿No? Bueno, entonces vamos a seguir. Les recomiendo de leer este tipo de artículo. No es el único a proponer guidelines y todo eso, pero vemos que, no sé, los 2 últimos años hubo más y más, tipo el artículo de Lucy también porque vimos un montón de publicación, de ensayos, y ahora yo lo veo también con mi publicación. Ahora hago mucho menos errores que tipo 5 años atrás. Así que, bueno, hay que saber un poco más. Bueno, volvemos. So back to our experiments. So I think like for this step, like the sample collection, we have discussed about, a bit about the expected and non-expected taxa, but we have discussed a lot about the reference database, a little about the use of pilot experiments, a little about the discussion about biological replicates, but which is not proper to eDNA. We have not discussed that much about field negative controls, but it will depend a lot about your sample also. But I think we are kind of ready to go to the other steps, which is kind of important at one point. One thing I didn't develop, because it really depends on the group you're studying, it's to know that you can have a lot of effect of the choice of primers. So for example, for fungi, it's obvious there are debates on the primers you would use, because some are more specific than others and so on. So for this, we could discuss it here, but for today it may be a bit short, but I think that's maybe something to develop in your proposal in the small project, to be sure that you know your primers, what are their limits. Just to give you an example, for example, you saw this morning these graphs about the ectomycorrhizal fungi. I was searching into the roots in French Guiana. I showed also that a lot of fungi were unknown. I didn't speak about this class, the endomycorrhizal fungi, the AM. Indeed, my primers, I use the ITS1 primers, the ITS1 region with different primers. My primers are allowing to sequence some glomerulomycota, but I know they are biased. I know that I have big genus of glomerulomycota that are not well amplified. I'm not, but however, for glomerulomycota, it's a big mess if you somehow working on it. For example, the 18S are good, but some genus are also underestimated or not sequenced. ITS2 is also discussed, but some are not sequenced. Normally in that course, there was someone working on orchids. I never saw him connected, but the idea is that on orchids, for example, the fungi specific to orchids, the Tulasnelaceae, are really not amplified with the ITS1 or ITS2 region. So indeed, if you know your primers, you know, okay, depending on the target I have, I may adapt the choice of primers. And it would be the same question for Sanger and for eDNA. So that's why I did not develop. So you have to know it, that's important. And if you have to choose, that's something Lucy and also presented. You have to read articles first to know what the errors of the others. Eventually test in silico. So in silico approach, you can still try to test, download some species from GenBank, check if your primers are aligned with, and so on. And eventually test in vivo or test in PCR and make your own test. The problem with the tests, as I mentioned to Thomas, is that also it costs a lot with eDNA. So often we try to make more tests in silico than tests on PCR. Okay, so I thought about a break, but I think we discussed enough. So we'll skip the break if you're okay, to finish a bit earlier. To finish today, so I wanted to finish with the a priori knowledge we need for Monday. So I wanted to discuss a bit about threshold and the choices we have to do during the analysis. So I think we are done for going to analysis. So so we know doing the analysis. Indeed, we'll see that during the analysis we have a lot of thresholds and choices to make. So we could wonder why. For example, when we see for example, with the ASV during the practical of Alexis Carteron, we saw that indeed we can have different approach, not necessarily threshold. But on the other hand, it's good because it's really reducing the time in analysis when you're using when you're handling a high number of sequences. That's the same thing that applies, for example, this morning I showed you that for a few sequences, you can make phylogenies, go to Papara, search for your sequence, but you cannot do it for millions of sequences. It's infeasible. So on one hand, we have like more sequences, more sensitivity, more species coverage and so on. But on the other hand, we have so many errors, chimeras, bias to reduce that threshold are really useful to sort. Like often it's a bit severe. And that's how, for example, the article I presented before was like having different threshold, strict or like relaxed. So we can have different ways. The real challenge is to reduce time in analysis and be able to sort numerous data and keep maintaining a biological sense also. So to give you an example of the threshold, indeed, that's a nice exercise to do also. I picked up an article we recently published with Julian Donald. So, and I highlighted just the way where we had some threshold used. So, for example, sequencing was conducted on different Illumina platforms, depending on the market considered, using paired N technology. Paired N, that's good because it means we should have two versions of the same sequences in two senses. Bioinformatic analysis of generated sequences were performed on the GenoTool platform, so we'll use it on Monday. With the OBTools package, same thing, use it. Paired N reads were assembled, primers and tags on each read were removed. So that's one point, being assigned to their respective samples. So that's one step important. Sequences were dereplicated and those of low quality or represented by only one read were removed. So it means we'll dereplicated, it means we keep only one represented per sequence. We'll remove the low quality, the singletons, and we'll make OTUs using a threshold for eukaryotes, fungi, and bacteria. And for example, using a different threshold for plants. So what do we learn from this? We know that, okay, we have two versions of the same things, of the same sequence. Okay, great. We'll dereplicate, so we will have even more versions of the same sequences, so we'll have some variability. We may have filters on variability. Then we have filters on the low quality, on the singletons, and on the threshold. So I will detail this. Okay. I will use for this perhaps, oh no, I will discuss it later, but anyway, here, just to make the link also with what we'll see later and what you saw yesterday. Here is, for example, a figure from Data2 pipeline from Alexis Carteron. So what we saw is that we may have, okay, here there is a quality check. So quality, okay, we'll use it. We may filter and trim, that's removing the primers, same thing. We may have some error learning, that's how we take into account the variation, so through dereplication and so on. We may remove the, also at one point we may remove some singletons, and here it's dereplicated and removing some singletons. We make also some clusters or OSV inference. So that's a way also to take into account the variation. And then after, there are other ways of the other important steps. We're merging. So here it's binding the two different versions of the sequence, R1 and R2, the forward and reverse. So indeed, that's also a step of filters. We may search for the chimera. We'll discuss about it. And finally, we use the taxonomy. So here is the pipeline for Data2. We saw that indeed it's using the variability, searching for unique sequences, searching for quality, searching for chimera, merging the two versions of a sequence, and using the taxonomy. So that's the different steps useful. In my case, I work with another package. So step by step, I will introduce you to another package, which is interesting also. You're not obliged, but it's good to know. And indeed, we'll see the different steps. So here it's my different steps I want to discuss with you. But finally, it's the same thing that for Data2. At one point, we made a check on read quality. We use the two versions of a sequence. We'll try to assign it. We'll have the filter on the quality. We'll reduce the singletons. We'll remove the chimera. So finally, you can have different pipelines. The answer is it's always the same, more or less the same steps that are used to filter the sequences. The idea is to reduce always your time for analysis and reduce the size. For example, we can shift from a single data set from 25 millions of sequence, or perhaps 1 million or 100,000 different sequences, but with a total abundance of 25 millions perhaps. But you want to reduce the number of sequences to be able to open your file at one point or to read your file. Anyway, so that's the objective of this Monday, is to know how to sort. So before this, I would like to be back on the few points where we can have filters. First, I wanted to know if you know what is a Q-thread. Do some of you have already make a filter on Q-thread? Perhaps not. So indeed, I think it was perhaps too early. To present you the Q-thread, I will first present you what is a sequence, what is a basic sequence in my sec, for example. So here it's a figure with a basic sequence. So a sequence has a name in general. It has a nucleotides, but you see it's often followed by a number of letters, of symbols, and so on. Have any of you already opened this kind of sequence or not? This is called the FASTQ format. Perhaps not. Bueno, entonces I will show you after, perhaps just to show you some basic. This is what, for example, a sequencer will send to you. Sequencer, you will receive like this huge file with all the sequences in it, and and it's called the FASTQ format. The good thing, indeed, if you first look at the symbol, there is the exactly same number of letters and symbols. And what does it mean? Each symbol here means a quality. For example, when you have this kind of mark points and I don't know the name of this symbol, it means the quality is low. When you have this kind of symbol, the quality is higher, and so on. The idea is that this is used by the sequencers, but you, in your analysis, you don't want this kind of information in your final table. So we'll try to resume indeed the different information. So what can we do to make a filter on the quality? In general, we use what we call the Q-thread. So it's a quality score, which is a probability of making errors all along the sequence. And it's used, so there is a Wikipedia page, quite nice indeed, it's describing the Q-thread for sequences. So I invite you to go to the Wikipedia page and read about this. The idea is it's kind of the best confidence interval you can have for Bayes. And me, for example, in all my analysis, I often make some filters on the quality of the score, and I choose 50 or 60. It's a bit high, but 50, like a quality score of 50, it means I'm kind of 100% sure of my Bayes. So it means I'm really I put a high threshold for my Bayes. If you think your quality score can be acceptable at a lower rate, you can of course decrease this rate. But it's important to know it, because at one point in the different pipelines, you often use this Q-thread. And for example, one of the functions I often use for my sequences is to ask. So I ask the cluster to compute, for example, an index. I mean, it has been already defined. It's not only me using it, but the idea is to compute the Q-thread for all the sequence, especially, for example, for the first 10 bases. So it means it's kind of the beginning of your sequence and the end of your base of your sequence. It's as if when you were, for example, using your Sanger sequences, you were looking at the primers. Okay, are my primers overexpressed? how is the tail of my sequence? That's the same thing. So this is the Q-thread quality. And you can also compute, for example, here I use a sequence quality like a mean for all my sequence. I divide by 250 because that's the length I expect for my quality score. But that's kind of my Q-thread for all the sequence. So in the OB tools package I use, indeed there are some functions that allow you to compute this index. The good thing is that you can annotate step by step your sequence with the quality number you would like. And so it allows you to reduce by half the size of your data set, because you will completely remove this part of information. So you will go from the FASTQ to the FASTA, and FASTA, instead of having always for each sequence all this length of information, you will squip it and then have only the number of Q-thread here. Okay, so that's an important score we will use. It was just to familiarize with you with this. Second filter we will use, indeed, it's about sequence length. So sequence length, it depends really about your marker. In general, for example, for fungi, I expect two pairs of 250 base pair. So I often make a filter at least for 100. So I would say like half of the sequence, that's a good half for us. It's always good to have a filter on the sequence length, because moreover with 100 base pair, you may not recognize any fungi at all. So, but that's something, there is no guideline for this, but it's important to know what's more or less what you expect as a length from articles, from in silico approach, whatever. But that's important to know it, because at one point you can make a filter, and that allows you to eject part of the full sequence chimera and so on. Another filter I use often, and indeed in the merging step of data 2, in the pair end step of OB tools, of as soon as you try to bind the R1, the forward and the reverse R2 sequence, you may have some filter on the pair end. So pair end, it means you bind your two sequences. For fungi, I always try to have like 50 base pair overlap between the two. So I ask, there is at one point a filter where you try to have your 50 base pair overlap. It's not obligatory. It can be longer, it can be shorter, depends of you. Again, it depends of your knowledge on your marker. So you can make, the good thing is that if you have a more overlap, you may be more secure about your sequence. And moreover, the more overlap you have, the more like accurate you are on all your sequence. So that's important. And finally, another filter we always used, it's about singletons. We always used, it's really common to remove the single sequences. Some are removing two sequences, some are removing more. So it depends a bit about what you expect. This singleton removal, it's like everywhere in all articles. It may remove a lot of sequences. Sometimes you lose like 5 million sequences. So that's super important, but it's important to do. And after, for the other sequences, often you don't need to remove it, because they may be just variants of PCR, so they may be integrated in your UTUs. So if I were using ASV, for example, like Alexis Carteron, I would perhaps remove more singletons, because I would say, okay, perhaps I have more variants. But it depends if they are used, but I think they are used to estimate the error rates. So they maybe you don't have to remove it. But in the case of UTUs, often they are integrated in this variation. So the best way to make your choices, indeed, it's perhaps if you have already some test pilot experiments, or if you have colleagues working on the same system, it's really to go back to the material and methods and read it carefully. See, okay, what were the filters? And you will see that some articles never mention any filters. So it's, wow, okay? Or perhaps we're really adding, for example, some threshold. For example, I know that some articles on fungi of perhaps or sometimes removing all sequences lower than 10 reads per count per sequences. So there are some really close other filters that can be used. And so the last filter I will discuss with you today is about the clustering methods. We already have discussed it a lot, but I think it's an important thing. So I won't be back on the debate about OTUs and ASV that we already discussed. And moreover, I think it's really, as I mentioned yesterday with Alexi, it really depends also on the markers of you're studying, the organisms, if you're studying population biology or community ecology and so on. But so just I wanted to have this small discussion with you anyway. Taking the example of fungi, of course, I wanted to be back on the choice of markers and then the clustering threshold we use. I will discuss about fungi and earthworms. So for fungi, if you're not familiar with this, it's a bit like for the organisms, but fungi are particularly known because they have several copies of what we called the internal transcribed spacer. Indeed, internal transcribed spacer, there is 1 and 2. So there will debate today about the use of 1 or 2. Why? Because this is already 300 base pair long. This is also 250 base pair long. So at the end, people have to choose between the MySec of this part or this one. As some of you know, we may one day go to long sequences and forget this debate. Anyway, the choice of this barcode is quite interesting. The good thing for fungi is that it's kind of pre-amplified. Like, for example, there are several copies of the ITS, like between 50 and more per fungi, per nucleus. So it means it's kind of pre-amplified. So that's why it was chosen as a barcode, because it's always easy to be amplified as in comparison to other nuclear part of the DNA. And moreover, it's flanked by like really conserved region, like the ribosomal DNA. So you have the SSU, the LSU that are flanked to this sequence, and that's really useful to be able to design primers. So we have variable sites and places for primers, and that's why it's so much used. Anyway, the other thing important to know with the barcodes is that, so we already discussed it a bit, but with ITS, we have kind of clear barcode gap. So it means the closer species are, the closer species share DNA like close to 100% or 98 or 97% are close. So we have a high percentage of similarity in comparison to interspecies or intergeneral comparison. It's due to highly variable sites, for example here, and it's due also to another phenomenon, which occurs during the speciation of fungi. I will discuss it later. The important thing about threshold, which is contributing to the debate, it's also that thresholds are kind of artificial. For example, here in this article of Joseph Gelb, he was comparing the species delimitation of Agaricus species in Alaska, in place where there is a huge diversity of Agaricus in the world. And he was showing the difference between the clustering at 95% and 98% for two different ways of clustering. Anyway, the important is not the way of clustering, it's always to show that, okay, at 95% similarity, we group things that are belonging to different species, so it's too low. And at 98% variability, we may be good for some species, not enough for some. So it may really depend on the species. Anyway, for fungi, there are no now some like common threshold used, but you always have to think it's kind of artificial. And if it's you're dealing with one known species, perhaps better to use phylogenies. So one kind of result I wanted to share. That's something away from eDNA, but I remember a discussion with Sébastien Gourbière about the speciation in fungi. This is, for example, an example of the DNA divergence between a population of, for example, a yeast, Saccharomyces, and so in the spore viability. So it means how much the crossing between two Saccharomyces would be viable. So it's kind of comparison of the biological concept of species towards the percent of divergence. And what we see is that as soon as you have more and more DNA divergence, at one point you begin to have a decrease in the spore viability. So it means at one point you have a quick accumulation of divergence due to like perhaps a biological barrier. And that's exactly what shapes the barcode gap. It's because at one point there is a like non-viable crossing that have accumulated a lot of variation in terms of divergence because it's no more under selection. And so this kind of DNA divergence, it can differ between, for example, yeast and here a bacteria. So the threshold might be different, especially because the speciation barrier might differ. And for example, within fungi, there is a huge variability, and some fungi are able to make fusion between different species. And so we can say that the biological barrier do not exist. So there may not be accumulation of divergence in relationship to biological species. But it's a discussion a bit away from eDNA, but it's always good to know, okay, why do we have a threshold and what's the biological sense of a threshold? Anyway, this really good review of Henrik Nelson a few years ago already, for example, reported all the thresholds used on ITS1 region, on ITS2 region. Lucy presented it a little. We see that indeed there is a huge variation. For example, there is finally, the amazing thing is that the conclusion was more about that we should use a 97% threshold for fungi. But if you see like a lot of fungi, like nearly half, or at least half of the studies reported like more 0 to 1% of variation differentiating within a species. So this is the intraspecies variability. So if you think about these results, we might increase the threshold for fungi, for example. But anyway, the 3% comes from other concept. Indeed, it's always a trade-off between intraspecific ITS variability and interspecific interspecies variability. So, for example, here we see that besides this huge variation in the level of intraspecies variability, like the mean is about 2.5, so that's why it's 3% which is used, like it's estimation, and especially because some species have a higher intraspecies variation. For example, some Basidiomycota, especially Glomerulomycota, the one that are able to fusion and so on. So indeed, finally, the threshold was a bit higher, and there is, I mean, everybody's using the 97%, but you have to know that like it's a big mean for, especially when you know that there are 3.8 million of species of fungi. Okay, and you saw this morning, moreover, that some fungi may have different copies of the ITS. So indeed, my results were kind of similar. Some species may have a higher variability indeed. So I checked the time. Just to, well, I won't be too long for this part, but just to show you some application, I had to handle recently a database on earthworms. Why? Because especially, so I showed you how interesting fungi were in French Guiana and how rare they are, and so on, blah, blah, blah. So basically I was involved in projects where we were reasoning not only on fungi, but only on earthworms with other groups of organisms, bacteria, trees, and so on. And we were always wondering about, okay, what's the effect of past anthropization on this kind of landscape. So you saw it a little this morning. I showed you the kind of sampling we did. We sampled also some habitats of earthworms, but the major response was this. So when I suggested to make this kind of curve, so here is the curve with eDNA on fungi. It's kind of exemplary. Here is the curve without eDNA, but with Sanger sequencing on specimen with earthworms. What we see is that we are not converging at all. We need more sample and so on. And so that's why we decided to shift to eDNA. The easiest way would be to use CO1. But I was not convinced, and my colleagues neither, because the database, the marker is really short, and well, anyway, the marker for earthworms are always short, but it was not that much resolutive. So we decided to go to 16S. But testing a new markers means we had to define also the threshold. So we had to search for the threshold used on 16S and know a bit what was the useful threshold. So for example, only from long sequences, we see that the intra-species variation was more about 10% so it was really higher than for fungi, for example. So anyway, the strategy, if you have to apply it also to your sequence, was to search for reference sequences with a new marker, 16S, know what was the variation in Sanger, also know what was the variation in MySec. We had a pilot experiment for this. We had to search in silico what was the threshold. And the last step, we were not still done on it, it's to check on specimens we know. So just to make this, there is one slide missing. But anyway, just to show you through an in silico approach for the intra-species variation, the intra-species variation, it's kind of huge in earthworms. We see that there is a majority of species that are extremely similar, but it may, we have also different samples that are said to belong to the same species that are extremely more dissimilar. From the intra-species variation, so there is one slide missing. We saw, because here it's on an experimental data set, but on Earth GenBank, it was the same. We saw that indeed, we better see the gap, the barcode gap, like around 93, 95%, 94% of variation. So we are still working on it, but it's the best thing to decide it was to make like the similarity, the histogram of similarity, and check again the specimens. So for example, now we are playing with the resolution, and we see that that's true that depending on the rank, you may recognize different family, different genus, different species. So you really have to know well your data set to decide the threshold. That's all the thing I wanted to conclude, because I see time passing. So for the threshold, the best thing you have to know is to see what was already applied, perhaps test. You can easily download, for example, a GenBank dataset and try to search for your threshold by yourself with an in silico approach. So that's a way to decide a bit the threshold. So for next week, indeed, we will go toward the different steps. Indeed, we won't wait. Going to the different step would be 3 weeks, but we'll do everything in one afternoon, because indeed it's short to explain each step. It's just the calculation time, which is extremely long. So I think it's feasible. The idea is we've seen the major filters. If we had other filters to add, that would be on controls, things that are more a posteriori and that are not necessary for the analysis of Monday. So what I suggest, I think I'm kind of done. The only thing that's it. The thing I've prepared for you, if you have time, I've put in the drive, in the data, I've put an example of data set of community ecology extracted from the pipeline I used on OBTools. I've put a small ones, so you can open it on your computer. You can have a look. I thought it was nice to have a look at what kind of data we can expect what is the aim. So that would be the end of an analysis. So if you want to have a look, you can. I mean, it would be a good way to prepare the next Monday. I will